I'm a little old, or you're a little quiet. You shouldn't be quite so timid. Howdy! Howdy! That's better. Okay, my name is Dave Bergbrider. I've been a professor here for almost 40 years, and they asked me to introduce Dr. Romo. I'm going to first start by telling you a little bit about the program, and the first slide illustrates some of the diversity of talks that we typically have during a semester. I know that one of the talks with Professor Shakashiri was canceled this semester, but we often have a number of people representing a variety of different areas of chemistry in a program that's generously funded both by the College of Science, and that's the picture of the dean up there in case you've never met him, and by Cengage, publisher of scientific books. The program also is very much indebted to Dow for support of the laboratory stockroom, and they also provide expenses for this lecture program that you're going to participate in this evening. The next slide introduces Professor Romo, but since I've been here for a long time, they chose me to say something about him. Now, most of you haven't gone to very many academic lectures, and normally at an academic lecture what we do is we talk about the person's CV, the curriculum vitae that talks about all the papers they've published and all the awards they got. And I could tell you things about Dr. Romo. I could say that he's about his career that he's had here since he finished a postdoctoral work at Harvard in 1993 and started as an assistant professor here. I could tell you about his research with beta-lactones that has led to a variety of things, but he'll probably say something about that. And most of you don't know what the hell a beta-lactone is anyway, so that wouldn't do much good. So instead, what I'm going to tell you about is some personal observations about Professor Romo. He actually is an Aggie. He was in the class of 1986. And so he's very much like you. He started here as a freshman, took freshman chemistry. I know this because in the spring of his sophomore year, he took sophomore organic chemistry with me. It was the chemistry majors course, and I took the time to go back and dig out some of the records. Now, that was a long time ago. I mean, I'm old, he's not quite as old, but he's still somewhat old. Back then we had these things called grade books where we actually recorded everybody's records. And Romo gave me permission to talk about his records, so I'm not violating the FYP Act. And I can tell you by looking at his grade book that 3 of the first 8 problem sets he got zeros, which isn't very suggestive of excellence in an organic chemist. But he did very, very well indeed on the final. In fact, beat everybody else by at least 20 points. And I think that was a harbinger of this career he's had where he's gone off and got a variety of national awards, given lectures on almost every continent except perhaps Antarctica and probably not Africa either. His background is he is a native Texan. I did have to ask him where he grew up. He said he grew up in a West Side barrio in San Antonio, lived there till about the 5th grade, and then moved to the Northwest Side, which I don't know I don't know much about San Antonio, so I don't know if that's a better or worse part of town. In any case, he's had a story. He's been very successful. He has 4 kids, 4 boys, one of which I think his second oldest is actually thinking of being or hoping to be a member of the class of, what the hell would it be, 2000, what class are you? Okay, he's going to be at least a year or 2 after you guys. In any case, that's, I think, enough, some background about Professor Romo. If you're interested, he actually does teach organic chemistry, and some of you will have the fortune, or perhaps sometimes misfortune, of taking organic chemistry, but he sometimes teaches the sophomore level course, and you would have an opportunity to, or you could have an opportunity to take his course and see if you can do better than he did on at least the problem sets. With that, I think I'll invite Professor Romo out to the stage.
Sophomore, that is not right.
Yes, it is.
Go sit down. All right, just forget what he said. I looked at that, and it was actually 10 out of 10. I don't know why he said 0, but anyway, howdy, Ags. Howdy. Got a little story to tell you, Ags. Actually, it's a long story. So what I thought I'd do today is tell you about some of the chemistry that has really, I guess, motivated me to do research. And this all pertains to a class of compounds that hopefully you guys will get to know here as natural products. And I have a lot of explanation to do in terms of the title, of course, because SpongeBob, cancer, obviously you guys know about cancer, but molecular engineering, what exactly is that? So I will hopefully, through the course of this talk, be able to define these terms for you and give you an idea of why I chose such a topic. So let me begin by making sure that everybody is up to speed in terms of the structures I'm going to show you. I'm going to show you a lot of structures today. So I really want you guys to understand what I mean when I show you something that looks like a hexagon, right? This looks like a hexagon and there's an extra line down here. Well, that's cyclohexene, right? This is a representation of a molecule that has 6 carbon atoms and has the complementary number of hydrogens to fulfill the tetrahedral nature of carbon, or in this case where you have a double bond, this has a slightly different geometry at carbon. So let me give you some more information here about what I will do in terms of the overall talk, and then we'll come back to what this structure exactly means. So again, molecular engineering, I have that in my title. This is really a playful word I gave to the term organic synthesis, because I really do see that what we do in the laboratory in many ways is a type of engineering where we're actually working at the molecular level. And so I'll try to explain more about that and what I mean by that as we go along. Again, I mentioned that natural products will be a sort of focal point of this talk, and I'll give you an example of a very complex natural product that I'll say a little bit more about, but I'll give you some case studies of natural products. Then I'll move on to show you some of the work that my group has done in this area of natural products, and also making derivatives of these compounds. I will show you some examples of how we are motivated by this idea in this very important area of synthetic efficiency. It could be said today that organic chemists, synthetic chemists can make just about any compound that we could dream up, but the issue now is how well can we make it? So it's not a matter of can we make it, it's how well can we do it? Can we do it efficiently? I'll tell you about some biosynthetic studies. These are studies aimed at trying to understand how these natural products, these compounds isolated from natural sources, are actually produced by the producing organism. Okay, so this is what I mean by biosynthetic studies. And then I'll show you how we actually make use of these natural products as probes to interrogate the cell. I'll then, again, give you an example and certainly try to explain why SpongeBob is at center here. Actually, it's because he's an Aggie. I don't know if you guys knew that. See that? Yeah. So I'll make sure that I explain that a little further. Then I'll move on to try to describe some of the studies that we've done with a particular compound, and I'll give it away now. This compound comes from a marine sponge, okay, and that's why SpongeBob is here. But the cellular mode of action, that is trying to understand exactly what this compound is doing inside of cells, and I'll try to end here by showing you the potential that the compound that I'll tell you about towards the end of this talk, what its potential is for drug development for cancer, and then I'll try to leave you with a few thoughts about the future of natural products. All right, so again, let me get back to these Kekulé line bond structures. These are one of the first things you learn. Maybe you've actually already seen these in some of your courses, and 101 or 102. And the idea, of course, is that, again, this is a representation of a molecule that maybe you have seen more like this. Maybe even back in high school you saw molecules like this that had carbons with hydrogens attached. But again, this is a shorthand notation, so you'll be seeing a lot of these. This is another way to represent this structure where now we're not just looking in 2 dimensions, we're actually looking at 3-dimensional version of this molecule. So we get an idea, a perspective of what this molecule might look like. And then a ball and stick model is another representation where you can start to see a little more features about what this molecule might actually look like at the atomic level. And then I think one of my favorites is this space filling model, where you can actually get a sense of the electronic cloud and how much space this molecule takes up. All right, so that's my introduction for these compounds, again, these structures that you'll be seeing a lot of today. But what is organic synthesis? So I need to really start by trying to describe what that is. So it's a branch of chemical synthesis concerned with the construction of carbon-carbon bonds, primarily. Okay, that's organic. And also included there are CH and C heteroatom bonds, like oxygen, nitrogen, halogens. But I took out this movie, and it's not very good quality here, maybe because it comes from here, but no, not really. Brent Iverson's a good guy, he's a good colleague. But I wanted to just give you an idea of what an organic reaction might look like if we could see it, right? So let me see if this hopefully will work. Yes, you can see a carbon-carbon bond is made right there, and then some other things are happening, and you get to a molecule, okay? So in reality, this is what we're trying to do in a flask, okay, in the laboratory, is trying to get reactants to come together and make new carbon-carbon bonds. So why do I call this molecular engineering? Let's see if this comes up. So, in fact, we do a lot of planning in our synthesis. We actually have to bring in raw materials, starting materials. We have to think about how we're going to work together as a team. Sometimes we do some pretty precarious things in terms of the types of reactions we do. We try to avoid those, okay, but sometimes we have to. And again, lots of teamwork and redesign of our initial thoughts usually don't pan out, so we actually have to redesign often our approach for making a particular molecule. So this is why I call this molecular engineering. And I really want to get across tonight, hopefully, that this is really an enabling science, in that if you can prepare compounds, if you can synthesize compounds, you're able to do a number of things in science. Okay, so to take a step way back in history, this is a reaction that was done by Friedrich Wöhler in 1828. And this is a reaction that was done at a time when everybody thought that organic compounds, that is, compounds that come from living organisms, could actually not be produced other than by an organism. Okay, that is, through the biosynthetic mechanisms of a living organism. So Friedrich Wöhler was able to show, again in a flask, that in fact he could prepare urea. Okay, and this is the first example of a reaction now scheme that allows you to get to a compound that was considered to be organic. And this is an important reaction because historically it also debunked the idea of something called the vitalist theory, which is what I just described, the idea that organic compounds could only come from living organisms. So it's a very important milestone in the area of chemical synthesis, and in particular organic synthesis. All right, so what are natural products? I mentioned already these are compounds isolated from natural sources, and I try to show you some of the sources that these natural products come from. It could be plants, it could be marine organisms. But what isolation chemists are people that actually go and identify and collect these types of sources of natural products. They grind them up, and they get a whole mishmash of compounds, okay? Great mixtures of compounds. They have these so-called crude extracts, where they extract these sources of natural products. And then they use something called bioassay-driven isolation, because typically these isolation chemists are interested in finding biological activity. That is, they want to find compounds that have some utility, maybe for animal or for, importantly, human health. And so these are some of the assays that might be used. If you're interested in cytotoxic activity, this might be towards anti-cancer activity, immunosuppressive for immunosuppressive drugs, basically for transplant rejection therapy, for example. And ultimately, you can get to single compounds that have this activity, okay? Now, this is a lot to show on one slide. In some cases, this whole process in going from the source to actually identifying a structure of an organic compound can be up to 10 years. Okay, so this is a pretty long, laborious process, and it's important to keep that in mind. And what I really want to show you on the next slide is something that really just, I guess it somewhat irritates me, because Hollywood, and I don't know if any of you guys are destined to Hollywood, but if you are, please listen, okay? So here's a movie. How many have seen this? I'm just curious. Oh, a few people have. It's amazing. It's an older movie. But I want you to notice three things. They're going to talk about something called a chromatograph. This is taking place in the jungle. I have to give you a little backdrop here. In the jungle, Medicine Man is about Sean Connery actually trying to identify some interesting anti-cancer agent that the natives told him about, and he's trying to figure out where it is, where it came from, and obviously ultimately try to get the compound and know its structure and hopefully help mankind. Okay, besides the poor acting, three things to notice. One is that they mentioned something about a chromatograph, again, coming into the I guess it's the rainforest. And so the question is, how do you get a chromatograph there? And do you have the right power supply? But besides that, they talk about how easy it is to go from injecting a sample into some chromatograph that they don't describe what it is, and you'll see what happens after that. So let me just get this started.
You were lucky you got me in last year's chromatograph.
Oh, Marge. Continue.
Okay. We're looking at 49 compounds.
Okay, 49 compounds. Okay, let's see where they go from there.
Okay, what we've got here is, where was it? Okay, 49 compounds. No.
Sodium
Nope.
phosphate, sodium
Nearly
nitrate.
all of these are identifiable. In short, nothing new. Is this why you wanted the chromatograph?
You said nearly all identifiable.
Yeah. Just this one, peak 37. Looks like an acid derivative. Although
If it was
those
only that
sites,
easy.
you must have really thought you were onto something. I'm sorry.
Can it be synthesized?
This one's Mother Nature's kitchen. End of story.
Okay, so, no laughing? Come on. I was the only one laughing up here, I guess. Maybe my colleagues down there. So, the fact at the end there where he says, can it be synthesized? He said no. She said no, absolutely not. This is Mother Nature's kitchen, end of story. What I hope to show you today is that, in fact, molecules like that can be made. Now, going back and what she said as well is that, you notice she picked the peak and kazoom, a molecule appeared. Well, it doesn't happen that way, okay? The structure of the molecule. So as I just told you, it takes sometimes up to 10 years to get to the structure of a natural product. All right, so let's get past that. So let me show you now two case studies, again, not from my laboratory now. These are historically very profound examples of how natural products have impacted the world. And let me begin with this very well-known example of penicillin. And so this is also a very interesting example from the standpoint that Alexander Fleming really has a great example of serendipity, because some of you may know that, in fact, the way he identified penicillin the very first time was by accident. That's a serendipitous finding. And what he found is, as he was getting ready to wash his dishes, he noticed that one of this is not his actual agar plate, but an agar plate actually had something like this, where he was of interest was the bacteria that he had streaked onto the plate, but there was another bacteria growing, and in fact you could see there's sort of a inhibition zone around this other bacteria that was growing. And so this suggested to him that whatever this thing was producing was somehow either killing or warding off the bacteria that would be growing here normally. And so that, of course, intrigued him, and this is, of course, the beginning, the serendipitous finding that led to the discovery of penicillin. So later, in the early 1930s, Florey and Chain at Oxford University in the UK, they were able to improve the fermentation and the extraction process. And then this actually became a very profound, again, project in terms of the ability to fight off infection. And in particular, in the case of in the 1940s, when World War II was just starting, this became an important collaboration between the UK and America in terms of trying to identify what this active component was that was leading to this ability to kill bacteria. And it was actually a worker at the USDA. The USDA was actually tasked with the goal of trying to identify this compound, ferment it, extract it, get it pure. And at that point also, they were trying to find a source of bacteria, a particular bacterium that would produce this in very large quantity. And it was this lady, Mrs. Hunt, at Peoria in Illinois, the USDA facility there, that actually brought in a moldy cantaloupe. And it turned out that moldy cantaloupe turned out to be one of the best producers because of the bacteria contained for penicillin. Other companies that you'll recognize, got involved in this huge effort to try to provide enough penicillin to support the war effort. And in 1944, in D-Day, when our U.S. troops stormed Normandy, it was very important that they had, in fact, 300 billion doses of penicillin ready. And the percent of casualties that would have occurred during World War II would have increased by 15 to 20% because of if we didn't have penicillin at that point. So you see some pretty dramatic things that came out in the newspaper at that point. So the structure was determined in 1945. So again, they started administering this compound without really knowing what the structure was. Here's some nice representations. Again, the Kekulé-line bond structure and a more of a tube-type structure of penicillin G, which was the first penicillin that was employed. Later, a total synthesis, not till 12 years later at MIT, Sheehan actually was able to synthesize this compound. Okay, so here comes in the role of synthesis in trying to not only access compounds that are natural, but also unnatural compounds to improve on their properties. So this is really a sub-discipline of organic synthesis, this idea of total synthesis. And what I mean by that is being able to access molecules like this, again, using the tools of organic synthesis, where you start with commercially available materials and try to move those and process those to complex structures such as penicillin. All right, so another very interesting example here is Taxol, and hopefully, and maybe some of you have heard of Taxol. This is a natural product that was derived from the yew tree and has potent anti-cancer activity. In fact, in 1971, President Nixon at that point was very interested in trying to combat cancer, and he was responsible for passing this legislation to really, and now it's called the War on Cancer, but at that point it was called the National Cancer Act that started that war on cancer. This, again, the yew tree, very beautiful big trees that grow in the northwest side of the U.S. These are Taxus brevifolia, and the problem with this is that, of course, these trees are very slow growing, and so when they started collecting these, and this was the original collection back in 1962 by Barclay, he found this compound he actually did this extraction and this collection, and it was only until later that they were able to isolate the pure active. This was chemists, again, isolation chemists, Wall and Wani at the Research Triangle Institute in North Carolina. They were able to isolate Taxol, which is this structure shown here, very complex structures you can see on the left, and this is again a space filling model. So it wasn't until 1971 that they were actually able to elucidate the structure of this natural product. So again, coming back to this concern about, I guess my pointer, neither one is working now. There we go. Nope. Well, it's very, very dim. Okay, maybe there's another pointer I can get, but this one doesn't want to turn off. Okay, oops. Okay, so let me see if I can pick up here. So the yew tree, thank you, appreciate it. The yew tree, again, is a very slow-growing tree, and so one of the concerns with this extracting which actually comes from the bark, is the fact that you would actually start killing a lot of trees if you wanted to get more of this compound. And so let me go further and say something about the mechanism of action. This is a microtubule stabilizer. You guys may know that, if you've taken some biology, that microtubules are very important. They're almost like the girders of the cell. And so on the left here, you can see that these are actually fluorescence microscopy of cells. Again, the control here, and you can see the green is actually these are fluorescence markers on these microtubules. And typically in cells, these should be looking like tubules or long lines. What you see in the case of Taxol is they start to aggregate, and you don't see the long spindly type of structures that are found in the normal cells. So let me show you this little movie, because what it demonstrates, you'll see that microtubules are constantly, it's actually an equilibrium process where these microtubules are being formed, and they also undergo collapse, or they fall apart, and so they're constantly being built and rebuilt and also degraded. And so what happens here with Taxol is that you can see it's microtubules forming, and then this is the process of where it's starting to come apart. Taxol actually blocks that type of process, where it actually doesn't, the microtubules aren't able to come apart. So that becomes very important when the cells are undergoing mitosis, and actually they basically arrest cells, and in particular cancer cells, which are of course often undergoing mitosis much more rapidly than normal cells. So one of the problems here, as I mentioned, is the supply problem. The bark from one tree only led to one dose, okay? That's a pretty serious issue. And so this is where this idea of molecular engineering comes into play again, the idea of synthesis. And what Potier found in 1981 is that needles from this tree, okay, so just the saplings of this tree, are able to produce this precursor to Taxol on the left. That can then be converted to semi-synthetic Taxol, as it's called, because of the fact that it's not a complete total synthesis, it's a semi-synthesis, by a process that was developed by Holton at Florida State University. This actually made him a very wealthy person, because this conversion of this compound, which comes from the needles of the saplings, can be combined with this compound to get to Taxol. And Bristol Myers actually was in charge and were able to do this on much larger scale. They also found that the ornamental version of the Taxus brevifolia tree, the Taxus baccata, actually was also a very good supply, provided a nice supply of this precursor. So in 4 additional steps, they're able to get to semi-synthesis of Taxol. Total synthesis of this compound took place much later. 1994, 2 syntheses came out basically back-to-back, Casey Nicolau and Bob Holton at FSU, and later many others that completed the synthesis of this natural product. So let me tell you a little bit more about why it is important that we complete natural product total syntheses. And historically, as I haven't really given you an example of this, but there are many cases where the structure of the natural product is not easily determined unless you can get X-ray analysis. Okay, but if you don't have access to crystalline material derived from the natural product, you can often use synthesis, and historically synthesis was used to determine the structure. The other thing that's very important is that if you have access to the natural product, you can also think about the fact that that will allow access not only to the natural product itself, but also derivatives. And importantly, design derivatives that might have improved solubility or potencies. And again, as I mentioned, we're interested in, and many are interested in, understanding the mechanism of action. That is, what are these compounds actually doing at the molecular level inside cells? So that's what I mean by mode of action. So you can prepare synthesized cellular probes derived from those natural products. You can impact biosynthetic studies, as I'll show you. One interesting example that has come out of the labs of ASI is a compound that was recently approved by the FDA. This is a pretty monumental synthesis because it actually takes 45 synthetic steps to prepare this compound, Halovin. And here's the formulation of that. But a 45 synthetic steps synthesis actually is pretty unheard of in terms of a drug that has been developed. Typically, fermentation in combination with synthesis, or as I showed you, semi-synthesis is typically employed to access natural products. And in this case, this is an anti-cancer agent that has very potent activity. The other thing that I'll mention here just briefly is the fact that often synthesis of natural products, or a desire to synthesize these natural products, will often inspire us to think about ways to improve synthetic efficiency, which is something I mentioned early on. So here's an example from my own laboratory. Some of my students, in particular Carolyn Leverett, as well as Vikram Purohit, as well as Andrea Matla, and more recently others in my group have got involved in this type of reaction. And what I want to show you here is obviously these space-filling molecules, which show you, I guess, give you a better appreciation for how we can go and improve the complexity of these molecules in a single synthetic step. We go from a monocyclic compound to a tricyclic compound. And then one additional step, we can completely rearrange the morphology or the topology of these molecules, as you can see again from these space-filling models. What's actually going on here is pretty interesting and complex, but basically there's 2 bonds that are basically changing positions, and that leads to a very different structure in terms of the final compound that I'm showing you here. So again, this is an example of how in our search for syntheses of natural products, this often will inspire us to think about efficiency and also different strategies for making particular types of molecules. So now let's get to SpongeBob, because that's why you guys are here, right? Especially since he's an Aggie. I mean, look at that. So SpongeBob is here because, again, I've already mentioned that one of the primary interests in my group has natural products, and one of the primary sources of the natural products that we have been interested in over the past 20 years or so have been derived from marine sources, and in particular sponges. And so obviously the sponges that we're interested in don't typically look like that, they look like this. Okay, that's an actual sponge from the depths of the ocean. And these, of course, can give rise to natural products. Again, I've already showed you how organic synthesis can enable access to potential drug leads and ultimately, hopefully, to cancer. And I'll try to give you some examples of that. Okay, so I mentioned biosynthetic studies. This is the idea now where we're trying to understand how the organism, in this case, let's say a sponge, actually takes simple molecules and converts them to more complex molecules. And this is an area of research that we've gotten interested in. This is, of course, there's other people in the department, such as Ty Bagley, who studies biosynthesis as one of his main topics of interest. Here, we became interested in trying to figure out ways to make compounds that would allow us to figure out these biosynthetic pathways. So let me start with a hypothesis here that had been in the literature for some time, namely that this compound that I'm showing you in the middle, called a rhodin, okay, was in fact proposed maybe for about 10 years to be the precursor to all the natural products I'm showing you on the peripheral here. These are compounds that have been isolated by various isolation chemists, and all of them had proposed some way to convert this compound to all these compounds again on the peripheral. So this was an interesting hypothesis, and in fact, most chemists can look at this molecule and think about how this can be converted to these various compounds, okay? It's obviously not straightforward to you guys, but we are very interested in looking at these type of interconnections between simple precursors that are found in, say, sponges, and then we find these more complex molecules, and we're very interested in trying to understand the interconnection here between these simplified, or I should say these simpler starting materials, and these more complex compounds. Well, we thought we could actually address this question. This is proposed to be the precursor to these molecules. Can we, in fact, answer that to some extent? And so what we decided to do is use our synthetic expertise here to prepare a version of that molecule that now has labels on it. Okay, in this particular case, it has a nitrogen 15 label. Okay, that's a way for us to track then basically the fate of this molecule. The other thing we did is to replace these bromines, which typically these molecules contain bromines, you might have noticed in the previous slide. We decided to replace them with chlorine because we thought maybe this would have a similar, let's say, activity or potential utility to the enzymes that convert this to more complex natural products. So we did both things. We actually used either N15 labeled rhodin or chlorinated versions of a rhodin. And this is a collaboration that we had with Ted Molinsky at UC San Diego. And again, this was our goal here. And what we found, in fact, when Ted Molinsky was able to take sponges like this one, which again is a sponge that's known to contain some very complex versions of this molecule, he was able to grind this up and basically extract the enzymes present in the sponge. And he was able to show that, in fact, this compound is indeed converted to these more complex natural products that had been isolated previously. Of course, these are now either N15 labeled or chlorinated versions of those natural products that had been isolated previously. So this provides some evidence for this hypothesis that, in fact, this is, in fact, the precursor to these more complex marine natural products. Okay, so one of the other things that we like to do with biosynthesis in my laboratory is to, again, think about how this organism might be producing these compounds and use that as inspiration for how we might try to synthesize these compounds in the laboratory. So the question is, can we in fact mimic the capabilities of SpongeBob in the lab? So what does that mean? So here's one compound that we became very interested in about maybe four or 5 years ago now, agostatin A. This is a tetracyclic structure, again showing you now in this case a movie to just kind of give you a better appreciation for the staircase sort of look to this molecule. Very interesting structure in terms of the number of heteroatoms, lots of nitrogens, lots of oxygens relative to carbon. And importantly for us was the fact that it had very potent cytotoxicity to a variety of cancer cell lines, as low as 97 nanomolar in some cases. So this became a synthetic target of interest. So we then started thinking about how to synthesize this molecule in the laboratory. So what we do as synthetic chemists is try to find where people have actually isolated compounds that are related to the target of interest. So I show you two compounds here, cyclorhodin and agelamide J. And you notice I've labeled these rings here, and you can see that there's some analogy here between these structures on the bottom, left and right here, and the target compound agelstatin A. This gives us clues as to how this organism or various organisms are producing agelstatin A. Okay, so this became sort of our initial blueprint, if you will, for how we might go about synthesizing this. So here I'm going to show you something that synthetic chemists use quite a bit, something called retrosynthesis. Okay, this is thinking backwards. And actually, you guys should realize that this is actually a very good way to solve a lot of problems. So for example, how many of you guys have actually done one of these? How many of you guys have cheated and started down here? Yeah, okay. And it's because it's actually easier to do when you're working backwards, right? So the idea here is that's actually what we do in the laboratory. We start with the target and work backwards to try to get to simpler compounds that we can envision either buying or preparing very easily. Okay, so this is the process of retrosynthesis that we apply to this molecule. And again, with the idea of trying to mimic what the sponge in this case was doing in terms of making this compound, guided by those other compounds that were isolated, we came up with this retrosynthesis for this molecule. Something that you guys will see if you take organic chemistry or what I'm showing you down here. These are mechanism arrows, but they propose what we envisioned in terms of this molecule ultimately being converted to the compound here, okay, which as you can see is very close to the target. Okay, the only thing that needs to be done is add water across that carbon-carbon double bond. But we envisioned this mechanism to access agelstatin A. So my student J.C. Reyes, who was a fresh, young, starting graduate student, you know, very impressionable. I don't know if he's in the audience, but he decided to take this on, this challenge of trying to synthesize this molecule. And one of the things that he decided to do ultimately after he got to this target, and it took him some time to get to this intermediate, which are key intermediate to actually make this compound, okay? And this was our initial attempt. Actually, I shouldn't say it's our initial attempt. It was our initial strategy for actually making compounds related to agelstatin A. And we're very pleased, JC was very pleased to find that in fact he could make the first ring that we wanted to make in this particular process that I'm showing you here. So we take this compound, treat it with trifluoroacetic acid, relatively strong acid, either at low temperature, or we actually found that we could do this at room temperature, and within 5 minutes you could actually access a compound that really starts to look like agelstatin A. So he was pretty excited. What I want to show you here is something that really is not very usual for us as synthetic organic chemists, and that is that we are able to actually see color in this reaction, okay? This is not typical for organic compounds. They typically don't absorb in the visible region of the spectrum, electromagnetic spectrum, but in this case, and JC did some detective work using molecular modeling to actually figure out that in fact it was presumably this intermediate which is formed, and you can see that red color forming. This is the intermediate that's responsible for that red color, again through some modeling experiments that he did to try to calculate what wavelength this molecule would absorb. And in fact, this was something we published, and JC was able to, in fact, convert this ultimately to agelstatin in 2 other steps. We're able to get to the natural product of interest. Of course, JC is very pleased as he graduates and moves on to a job. So here, JC is not only good in the lab, he's also rather artistic. He came up with this nice cover that came out on a journal that we published in, Angewandte Chemie. Here's the overall synthesis. You can see the simple starting materials. JC was able to take this and convert it ultimately again to this agelstatin A compound, and now he's actually towards the end of his stay here in my laboratory, he's actually making use of his synthesis to prepare compounds that hopefully will allow us to figure out what this compound is doing inside human cells, in particular cancer cells. Because again, this is a very potent anti-cancer agent, it kills cancer cells. We would like to know how and why this compound is able to do that. So he's very interested in actually making very good progress in making probes to answer that question. This is a collaboration with Professor Jun Liu, a longtime collaborator in my laboratory who works at Johns Hopkins, and we've actually done a number of projects, and the last project I'll tell you about actually has to do with a very nice collaboration we've had with him for the past 10 years or so. So let me show you one more example. I'll go a little faster through this one since I'm a little short on time, but I think we're doing okay. These proteasome inhibitors, these are compounds that inhibit the proteasome. Let me go back one slide to simplify it. So lactocystin, amuralide, and selenosporamide. And Professor Bergreiter, when he introduced me, mentioned something about beta-lactones. So these are compounds that have 4 atoms in the ring. You can see that that's common to these molecules on the right. So there's an oxygen, a carbon, a carbon, a carbon, and then a carbon-oxygen double bond. That is a beta-lactone, and that is contained in the structure of amuralide and selenosporamide A. So we became very interested in these compounds. Again, we started thinking about how these might be synthesized, in this case by the bacteria that produces these compounds. Okay? Actually, this is derived from a terrestrial bacterium, and this is from a marine bacterium. So one of the things we thought is it would be very nice, again, in a retrosynthetic sense, working backwards, if we can start with these targets, we work backwards and say if we can break this bond and this bond in a retrosynthetic sense, in a reverse sense, we get to some compounds that are relatively simple and ones that we can envision we could prepare rather easily. So I will just show you that key reaction that we developed as a result of these natural products. Again, here's selenosporamide A. This is the compound that we're after here. This is the starting material. I drew it sort of in an extended form and then in a more wrapped up form that starts to look like the compound of interest. And in fact, in a particular reaction that we developed, we can in fact take this rather non-cyclic compound, as you can see it's non-cyclic, and you can actually make a bicyclic compound. So this is a bicyclization reaction that forms 2 rings, the 2 bonds I'm showing you there, in one step. We can do this on gram scale, reasonable yield here, to get to this target rather rapidly. And in 4 additional steps, we're able to make this particular natural product. So again, this is hopefully demonstrating to you the idea of trying to take clues from how organisms might be producing these compounds and using those to inform us in terms of how we might synthesize these in the laboratory. All right, so let me just finish by showing you a couple things here. One on the left here is the proteasome. This is a nice cartoon of the proteasome. The proteasome is basically the recycling bin of the cell. Okay, so when peptides are no longer needed, they basically spool into one side of this machine, and it basically chops up proteins into small peptides. Well, it turns out that's actually a very interesting target for cancer. There's actually now 2 drugs that have been FDA approved for inhibiting the proteasome as an approach for cancer, and these compounds are actually in phase 2 clinical studies, selenosporamide A in particular, for inhibition of the proteasome. And here show you the synthesis that we have as well as how this thing actually inhibits the proteasome. But one thing I wanted to show you was, again, another cover, because it really brings out this idea that I'm trying to get across to you, where we're trying to mimic what this organism does in the actual organism itself, how it synthesizes these molecules. I don't know how many of you guys can see this. Can you guys tell what's in the middle here? Can you guys see that? Looks like a sponge. I heard somebody say sponge. Any other ideas? Coral. I see. Yep, coral's there. Something in the middle here. Look, there's an eye. Seahorse. Good. Seahorse. Yep. So it's pretty cool, right? This is an example of biomimicry, where the seahorse has evolved over time, presumably, to mimic this coral that it's sitting on, right? So it makes it very hard for predators to find it. Well, this is exactly what we're trying to do in the lab. We're mimicking the biosynthesis of these molecules, similar to the way the seahorse has mimicked its background. Okay, so the story that I want to tell you here at the end is the story of this compound. This is a compound that we've been working on actually since I started here at A&M. We've made some progress, so this 20 years, you better hope we've made some progress, right? But this compound, patimine A, is derived from this yellow tubular structure that's behind this fish. I think the photographer is more interested in the fish than the sponge. But this sponge back here, this yellow tubular structure, is what gives rise to patimine A. This was isolated off the coast of New Zealand in 1991. And again, some very interesting cytotoxic activity, in particular towards leukemia cell line at 0.27 nanomolar. It's very potent compared to 0.54 micromolar, so a 2,000-fold differential between normal cells and cancer cells. That's something you, of course, want to see for any compound that might be of interest as a potential drug lead. And in this case, patimine A, that did demonstrate that type of activity and that differential. So one of the things that we wanted to do, of course, is to save SpongeBob, okay? Because, again, just like in the case of Taxol, in order to get lots of this compound, we would have to go and extract out all this sponge, and the environmentalists have some concerns and issues with that. So one of the reasons that we want to do total synthesis then is to, of course, not have to worry about harvesting lots of sponge. So we came up with a synthesis of this molecule that we published back in 1998. This is what's called a convergent synthesis, okay? So here's a retrosynthesis again. We start with the target, we go back to some simple starting materials. It's convergent because we actually come up with 3 main fragments that we envision could be joined at a late stage to construct the target of interest, okay? So we do that. This is the synthesis. These are the starting materials that give rise to each of these pieces. And this was a 24-step synthesis, okay? That's actually reasonably long, not something you want to be doing in terms of, large-scale production. One of the other things I mentioned early on is that total synthesis also often will confirm structure, and in this particular case, we're able to confirm the structure of this molecule through our total synthesis. Okay, so one of the things that we did, and I'm going to skip a lot of background information here, but we recognized that we probably could remove this group and this group highlighted in blue and still have a compound that might have similar bioactivity. Okay, so we decided to do that. Lots of details here, but I just want you to recognize that we now made a compound that's related to ptamine A, but now those 2 groups are missing, right? This actually simplifies the synthesis dramatically. We actually removed 10 synthetic steps. So now it's a 14-step synthesis. And fortunately, based on our hypothesis, this compound actually did turn out to be equipotent to the natural product. So this is a compound that was patented by Texas A&M, and we continue to try to develop this compound as a potential drug lead, and I'll show you some more examples of how we're doing that. So this next part is, once you have this natural product, you have access to the natural product, you'd like to figure out a way to use those compounds to try to figure out why does it kill cancer cells. This is a very interesting sort of basic cell biology type of question to ask. Once you have a small molecule that is killing cancer cells, you want to ask the question, how is it doing it? So to do that, the analogy of fishing is actually pretty good because you basically want to take the molecule of interest, in this case ptamine A, and basically make a fishing rod out of it. Okay? And that's to fish out the proteins that are interacting with it. So here's the synthesis. I'm not going to go through that. We got to a compound on the bottom here that actually has the structure of ptamine A here, but it also has this interesting handle here. Okay? Now, any of you guys who actually fish know that the way you hook a worm on the hook matters, right? So if you don't hook it well, the fish might actually take your worm and you never catch anything. It's the same thing with this type of approach here, where we need to figure out where the best place is to attach the hook, if you will, to the natural product of interest. And this took us some time, but we were able to figure out that this was basically the best type of molecule to make. So we sent this over to Jun Liu at Johns Hopkins, but I want to first tell you about how you construct this fishing rod. So you can buy beads made of agarose that actually have a protein called avidin attached to it. Okay, so this is basically our fishing rod. The bait is now this molecule biotin that has been attached to the natural product of interest, ptamine A. So now we have a complete fishing rod, and we're going to use that fishing rod to basically fish out, hopefully, some protein receptors that come from the cancer cells that this molecule is killing. Okay? So we want to find out what, in fact, are these proteins that are binding to this small molecule. So Jun Liu at Johns Hopkins was able to make use of this bait that we synthesized in the laboratory. And again, I'm not going to go into great detail about this, but there are bands here that you can see. And basically these bands represent proteins. Okay? This is an SDS page gel electrophoresis experiment. They can separate proteins. They can actually identify these proteins by a technique called mass spectrometry to actually figure out the weight of these proteins. Besides the weight, they can start degrading the protein and ultimately figure out what is the actual protein that is present in this band, again, that basically was caught by this bait. And doing that, they actually found this protein of interest, EIF4AI, okay, which actually stands for elongation initiation factor 4A. This is a protein that's actually important for protein synthesis. Okay? And what Jun Liu actually, through a number of experiments, again, that I'm not going into detail about, was able to show that when ptamine A, and you can look on the left picture here, actually these little red dots coming from this presumed sponge actually interact with the protein, and you can see that it's interacting, but ultimately this protein complex formed between the natural product and the protein are involved in protein synthesis. Okay, and what patimine A does is basically stop protein synthesis. So for cells that have been administered patimine A, you can basically shut down protein synthesis. And I'll tell you a little bit about how that is important for cancer and a potential treatment for cancer towards the end of the slides here, next couple of slides. But let me show you this movie because I think it really gets across this idea of how this molecule is exerting its anti-cancer activity. So basically, this is inside the nucleus. We just synthesized messenger RNA. And again, if you've taken biology or you're taking biology, you know that messenger RNA has to exit the nuclear pore. And messenger RNA that will then try to find a ribosome, and the ribosome is where protein synthesis takes place. And in fact, the ribosome, it consists of two big pieces. Here's one piece, here's another piece coming in. Patimine A stops the process before it even gets started. So basically, when the messenger RNA interacts with that first half of the ribosome, patimine A is bound and it prevents further protein synthesis and further prevents the process of protein synthesis. So this again has potential now for anti-cancer activity and thinking. There we go. And so let me try to get across what potential this has as an anti-cancer agent. So this is a pretty dramatic photo, and it's actually an image of a patient that has a type of cancer called inflammatory breast cancer. And the red spots, of course, are where there's a lot of cellular activity. And unfortunately, this particular type of cancer, again, inflammatory breast cancer, which is being studied by Dr. Robert Schneider, a collaborator of ours at NYU, this is a type of cancer that basically once it's been detected, it's almost too late. So there's really no good treatment for this type of cancer. And this is just, again, a very dramatic photo of showing where this cellular activity, it's already spread up into the neck, as you can see, and even lower below the breast. And it's a pretty dramatic photo to show that. So one of the things about cancer cells is that you may know that they're rapidly dividing. Okay, so cancer cells that are rapidly dividing are making use of a lot of DNA synthesis. There's angiogenesis that must take place. Actin and microtubules are actively being formed and degraded, as I showed you in a previous slide. And then this important area of protein synthesis, which is very important for a cell as it divides, it needs to generate new proteins. Now, the thing about cancer cells relative to normal cells is that they're actually in a very delicate situation if you just tilt the balance just a little bit in terms of slowing them down. They actually it'll lead to something called apoptosis, and this is called means programmed cell death. So basically, you don't have to tilt the balance very much, okay? If normal cells were affected by these compounds, okay, it takes much more to tip that balance, okay? But in the case of cancer cells, it's much easier to actually tip that balance toward apoptosis and ultimately then to cell death. So in the case of patimine A, again going back to inflammatory breast cancer, these are some results from Bob Schneider's laboratory, where he was comparing now, you can see the tumor size on the y-axis and the number of days of administration on the right, and also observation of, this is actually a mouse tumor model of inflammatory breast cancer. And you can see the control where there's either DMDA-PAD-A, which is that simplified version of patimine A, or radiation. So this is actually a combination therapy of radiation plus in this case, protein synthesis inhibitor. And you can see with the compound alone, there's some reduction in tumor size as days go on, but still there's pretty rapid growth. Again, this is a very aggressive cancer, and so it's very difficult to stop this cancer. Radiation therapy alone has a more dramatic effect, but you can see that after 35 days, there's still a pretty large tumor that's present. And of course, then what's most impressive here is at the bottom, where you combine now this protein synthesis inhibitor with radiation therapy, and you can see that there's almost a flat line in terms of growth of that tumor. So that's very good news in terms of the potential of this compound for treatment of inflammatory breast cancer. So we continue to look at the possibilities for this compound, again, for this cancer and several others. So let me leave you now, stop here by showing you what I think the future of natural products are and might be. One of the things that I'm showing you here is on the right, the target discovery information and functional genomics and lead discovery. So typically, the way drug development is done is that targets, so biologists have had a lot of limelight because they identify a particular target that is an enzyme target or a protein target, and they find out that that target is important for development of cancer. They then use information and functional genomics to figure out what is this target, what is it important for, and then they start screening small molecules, such as natural products, to identify small molecules that will inhibit that particular target. You then, once you have a lead or a hit, you then move on to what's called medicinal chemistry, where you start optimizing that structure for inhibiting that particular target. You then have to worry about the pharmacology of this molecule. What is its solubility in the blood serum? What is its clearance? Things like this. And ultimately, you get to hopefully some preclinical development, and much later you get to ultimately drug discovery. Okay, it's a very long process. This can take up to, again, maybe 10 years in this case as well, or more. But one of the things that natural products has been able to do is to, in fact, impact this way of doing things. And historically, this has been the case. We sort of had a decline in the interest in natural products, and now we have an increase in the interest in natural products. So one of the things that happened, of course, in 2003 was that the human genome was sequenced. And what they found, which was pretty profound, is that of the 3,000 to 10,000 proposed disease-relevant genes, okay, only maybe about 500 of those targets have actually been accessed. This is as of 2004. So what this tells you is that there's a number of protein targets that are still possible. And the question is, how do we get to these targets? Can we find small molecules that actually interact with these targets? And so natural products, I think, provide an interesting way to do this, because natural products actually are the lead compound. Because the idea is that if you have a natural product that has bioactivity, you know it's doing something in a cell. So that's actually your starting point. And you ask the question, what is this natural product doing? You use that to actually identify a target in the cell, and then you can continue the process as before. Okay, so it kind of turns everything on its head. Natural product becomes a starting point, and we use that to identify a target, like I showed you in the case of the protein synthesis inhibitor pteamine A. All right, I want to leave this last slide here to just show you that I'm very interested in undergraduate research. One of the things that I initiated in the past 2 or 3 years is this mini pharma, TAMU Undergraduate Mini Pharma. This is actually a collection of undergraduates working together, doing structural biology, biological assays, and synthetic chemistry. You can see some of the participants right now and also some of the past participants, the alumni, if you will. But they're very interested in trying to develop compounds that interact with the proteasome and fatty acid synthase. In the last slide here, I just want to thank my research group, and this is actually somewhat dated, but I wanted to show some of the students that were involved in the projects that I showed you, show you my collaborators here, the funding that makes all this research possible. This is not possible just on my own pocket. This actually comes from government agencies that support this. And finally, I definitely want to thank my family who really make this possible. Oh, here's my family. No, here's my family. And I blotted my face out because you already see my ugly face. So this is certainly a support group for my profession, my work, and so I just certainly want to acknowledge them. Finally, let me thank you for your kind attention.